Oh, I'm Eugenie Scott. I'm the president of the Bay Area Skeptics. Dr. Elisabeth Bick is a microbiologist whose PhD is from the University of Utrecht. She worked at Stanford University for 15 years as a researcher, leaving academic research for the private sector. She founded Microbiome Digest as a source of information on the microbiome and has a very active Twitter feed. Oh, this is the wrong introduction. It is the same woman. But I had so many more nifty things to say. 27,000 Twitter followers. This is impressive. I have far fewer than that. In a profile in The Scientist, she is described as having a second career as a data cop. She has spent an extraordinary amount of time reviewing scientific articles, resulting in the withdrawal of 22 papers in PLOS ONE alone. She will tell you more about this work and other misconduct in scientific papers as she discusses plagiarism, fabrication, and falsification. Please welcome Elisabeth Bick to her first Skeptical.
Much. Can you all hear me? All right. So today I'm going to take a little different twist than some of the previous talk. I'm going to talk about misconduct in science. So basically, if you came here trusting that every science paper in the world is a good paper, I'm going to tell you that that is not correct. I am a data cop. I'm looking for science papers that have signs for misconduct or that have other signs that they're just not very good papers. And so today I'm going to talk about science misconduct and a little bit about dubious science papers and science publishing. And I'm going to start off with making sure that you also know that most scientists are very honest people. Most scientists work really hard, report really honestly on their papers, on their results, and you can trust those. I'm here to talk about the very few bad apples. And as you can see here on this slide, science builds upon science. So every science paper, usually, or almost every science paper will refer to previous science papers in their introduction and then build upon those facts that have been found by others to find something new. So if a science publication, that is the building block of science, if a science publication is incorrect or has incorrect facts, then it's much harder to build science upon that. And it might actually lead to years and years of wasted effort by a grad student who built their whole career maybe on a science paper that was flawed. So although most scientists are honest and hardworking, scientists could be biased. Like we all tend to believe the results that show the thing we thought was true. If we believe the Earth is round and we do an experiment that actually hints at that the Earth is flat, we probably would not really believe that experiment because we already have a firm belief that the Earth is round. Just to give a very bad example maybe, but we are biased in what we think is true, and we tend to publish the things we think fit in that hypothesis. But scientific papers are often not very reproducible. If you try to replicate exactly the same data or the same experiment that another scientist did, you probably will not find the exact same results. I'm in biology, and you might have a different incubator or a different cell line or a different type of shaker, and all those things could influence the results. So science papers are very rarely reproducible, but in general, most science is sort of as a grander scheme, it is reproducible, just not those very details that another scientist has found. A scientist might also apply errors in statistics or models or have, I don't know, the wrong formula in their Excel spreadsheet, and which might lead to errors. And so those are all, examples of science papers that might not be very good, but they're still made with the best of intents. But today I'm going to talk a little bit more about sloppy science and actual forms of science misconduct. So why do scientists commit science misconduct? Why do they make intentional errors or change data intentionally? Well, you realize that most scientists are under enormous pressure to publish. So this cartoon says publish or perish, which is sort of the motto of science. If you don't publish, you're not going to make it as a scientist. So in this cartoon, a new scientist is welcomed to the team by maybe the dean or his PI, and the dean or his PI says, welcome to the team. Remember, if you follow the university motto, publish or perish, you'll do fine. But else, your career is probably not gonna last at this university. So this enormous pressure to publish, to have publications every year, or to have an X number of graduate students graduate from your lab every year, is unfortunately leading scientists to choose the wrong path and to publish results that have been fabricated or falsified. So I first want to have sort of a very dry slide on what the exact definition of science misconduct is. So the ORI, the Office of Research Integrity, is a US organization that has a very clear definition for science misconduct. They are the body that investigates scientists who have worked with NIH, for example, money or other government money and funds, and have committed misconduct. And so they investigate these cases. And they define science misconduct as, they have 3 different types of science misconduct that you can find on their website. So the first one is plagiarism, which is stealing of verbatim text or ideas from another person. Doesn't mean stealing ideas from a person in your lab or from yourself, like self-plagiarism, or basically text recycling in another paper using your own text, does not fall under the definition of science misconduct by ORI. Then there's falsification, which is manipulating or omitting data. Like you can see on the cartoon below, this scientist is in the lab, and he or she has 3 Petri dishes with some results of bacteria, but one of them doesn't quite fit his or her hypothesis, and they push it from the table and ignore that result. That is falsification, because you're changing the data. You're leaving out certain data, like an outlier, for example, that does not fit a person's hypothesis. And then maybe the worst kind of misconduct is fabrication, totally making up data of experiments that had never happened, or patients that did not exist. So fabrication and falsification can sometimes be overlapping, and it's sometimes hard to draw the line what is falsification of fabrication. ORI also makes it very clear what they do not consider research misconduct. That is honest errors or differences of opinion. Of course, one scientist might come up with an idea and the other scientist thinks that that paper cannot be true or simply is wrong, and that could result in a whole discussion about whether one scientist maybe thinks that the other paper should be retracted or is false or whatever. Honest opinion, or differences of opinion, do not count as science misconduct under this definition. So let's dive into the first example, plagiarism. On this cartoon, I'll read it to you. The PI says to the graduate student, I need you to do a presentation on the topic of plagiarism. If you don't have time to prepare anything, just steal something from the internet. So I stole this picture from the internet, which, is plagiarism, but I try at least to credit the cartoonist who drew this picture. So I'm stealing something, but I'm giving credit to the person who made it. And because I give credit and I, make sure that I don't pretend this is my own cartoon, then you could consider this not to be plagiarism. So I hope I'm following my own rules, because I don't want to be caught doing what I'm preaching about, of course. So I got very personally involved in plagiarism because I heard something on the radio about science plagiarism. And I thought, let's take a sentence of my own, a sentence from one of my papers, and put that between quotes in Google Scholar. And, only my own paper should come up, right? Well, there was another paper that came up that was published a couple of years later. And I was very angry. It's like, they stole my sentence. This is my sentence. How can you write my sentence and put that in your paper and claim it's yours? Now, that paper was consisting of a patchwork similar to what I'm showing in this slide a patchwork of stolen text from other science papers, sort of a Frankenstein monster of other people's text. The example I'm showing you here is a different one, but it got me completely obsessed with these type of things, because you search for one sentence in Google Scholar and another paper shows up that stole that sentence. You look into that paper that stole many other sentences, and one thing led to another. And I found this particular example, a review paper that used text from at least 10 different source papers. This author was not a very unexperienced researcher, not a person who was maybe struggling with English writing or writing their first scientific paper. No, this author had over 300 publications, so he was, a pretty experienced scientist. But somehow he chose other people's text to make his own patchwork and write a review paper. So I saw this and I was a little bit surprised that this happened. So I wrote to the editor-in-chief of this journal, and this paper was retracted. And so it still exists, you can still go online, but it has a little watermark retracted over it. And that tells the scientific community that it's no longer backed up by scientists. I found this other example in which at least 43 different sources were used to create this review paper. And I was, quite frankly, just running out of colors here. Because, how do you mark this? And it's, of course, a lot of work to make these reports. I can just yell, this is plagiarized, but I need to show some proof. So I have to put every little part of a sentence into Google Scholar and color-code it according to the source. So it's a lot. It's like evenings and evenings of work that I was doing during my time that I was still employed. And so it took me sometimes weeks to do one of these papers. In this particular case, I was not really counting on any luck in getting this paper retracted, because the last author of this paper happened to be the senior associate editor at this journal. So I had, here I am, little scientist, writing that this paper maybe is not completely correct, but it actually got retracted. So kudos to the journal of doing the right thing. And so I started doing this more and more, and I used mainly Google Scholar. There are expensive plagiarism checkers like Authenticate that a lot of universities use to check the theses of their students. But I cannot afford that, because you have to pay for every single paper, and that would cost me thousands of dollars, and I'm not funded. So I just did it by Google Scholar, copy-pasting text or into regular Google. And I just made up a sentence. Plagiarism is not allowed in scientific papers. If you put that between quotes, there's no results in Google whatsoever. So if you write a sentence, if you're now thinking, oh, shoot, maybe I did some plagiarism and Elise Bick is going to catch me and have my paper retracted. If you wrote your own sentence, it's unique. It's not going to be plagiarized or anything. It's your sentence. There's exceptions for things like definitions. Like probiotics are live organisms that confer, the health benefit to the host, something like that. There's definitions that, of course, you have to stick to. Usually they're put between quotes. That's not plagiarism. I'm talking about complete paragraphs that are just copy-pasted from another paper. That is plagiarism, and that is something that does not really serve science. On the other hand, you could also argue, well, it doesn't really harm science either, because it's just regurgitating text that another person has also wrote. So it's not really falsifying science, but of course it's not really nice for the person who actually wrote the sentence themselves. So my personal project, I did 80 of these papers that I analyzed. It took me I think 2 years or so. Reported them to journals, and 35 of these papers have been retracted, which is a very high percentage. But it was so much work that I decided to switch to something different, which was looking for image manipulation. But I'll talk about that a little bit later on in my presentation. I first wanted to share a couple of examples of you. So we've covered plagiarism. Wanted to share a couple of examples of the other two forms of science misconduct, which are falsification and fabrication. So a very famous and very relevant to this audience example of research falsification is Andrew Wakefield. And I don't know if most of you probably will have heard of this person. He was a doctor who published a paper in The Lancet in which he claimed that 12 children that he investigated that came to him after measles vaccination became autistic or had autistic characteristics. And he described the case as being 12 consecutive kids who had a measles vaccination and became, had autistic characteristic immediately after the vaccination. And this particular paper is the source of everybody you have met who claims that they don't want to have their children vaccinated because of this particular paper. But it turned out that this paper is falsified. He did see 12 kids, but several of them already had health problems that might be indicative for autism development later on before they got the vaccination. Some of these kids never developed autism. He also claimed that they had colitis, so like an inflammation of the gut. So he had colonoscopies done on these kids, and the results were completely normal because they did not have any complaints. He falsified the results of these colonoscopies in which it was found that they had a normal-looking gut, and he falsified and he said colitis. So a team that went into these papers and looked into it found all these falsifications of data. And finally, after a lot of discussion, the paper was retracted. But the damage was done, and we all know what happened after that. And so I think this is, for me personally, one of the reasons I'm fighting science misconduct, even though I might be too late and I might be the only person doing it, but I feel it's a fight worth fighting because these falsified papers can do so much harm on so many people later on. A second example is that of Haruko Obokata. She was a rising star in Japan at the Riken Institute, and she claimed that she could treat cells that were differentiated, like some blood cells or so, that she could treat them with a very simple acidic treatment, little dipping them in an acid bath, and suddenly they became pluripotent, so a cell that can become any other cell. So basically, she was saying that she could treat cells from any tissue with treating them with a little acid and making them like a stem cell again. And that sounded too good to be true, and it turned out it was too good to be true. She had 2 Nature papers back to back. Immediately after publishing that, like within a day, people were starting to talk online about, this sounds so crazy, like why did nobody else ever find this? If it was so easy to make a stem cell, then people would have done that already. And people were looking at her images and finding that certain images from her thesis, which was on a very different topic, looked extremely similar or identical to some of the figures in her Nature paper on this treatment. And people started to look more and more closely at these pictures, and they found photos that were manipulated and flipped and rotated and whatever. And there was more and more discussion, and this paper was retracted within months after, which is pretty quick for Nature. That's like a very large and kind of slow sometimes
journal. It got retracted in July, which was like 6 months or so later after the publication of these papers. Haruko was then given the opportunity to replicate her experiments by working under video surveillance for a year or so to replicate her experiments. She was never able to replicate any of these experiments. As a sad side story, her mentor, who was co-author on some of these papers, who was not involved and never has been suspected of misconduct, he committed suicide by hanging himself in the stairway of the Riken Institute. And I'm saying this because I'm very aware of the work that I'm doing, how damaging it can be for the careers, not only of the people who can do misconduct, but also of all the people in the labs and all their families who are involved in this. But I can also not look the other way and pretend I didn't see it. But yeah, it's very serious business to do this type of work. As an example of research fabrication, so the complete making up of data, I am sharing here the example of Diederik Stapel. He, like I am, he was Dutch. He's still Dutch, but he was a, I hope, although the Dutch people don't really embrace him as much as he did, but he was a very, very well-known psychologist who had very a research that was always in the newspapers. He did amazing data. He had amazing data sets, and but he was always very secretive about it. He claimed he had done research in schools, and he would give his graduate students data set that were obtained from these children or whoever he did his research on. But he never shared the raw data. It was already analyzed, and and he would give that data to his students, and they would work on it and write papers. But the students grew suspicious after a while because some of the data looked very weird. If you look at the numbers, like certain numbers were much more prevalent than you would expect by chance, or some of the data looked very similar to another paper. And so they started to observe him, and they finally thought, well, this data cannot be true. Like, he's just making up this data. And so another investigation was started, and it turned out that most of his data was indeed fabricated, so completely made up. He has written 137 scientific papers, according to a couple of sources, and 58 of these have been retracted so far because they were fabricated. He's pretty high up in the leaderboard of Retraction Watch, which is a site I love to follow. So Retraction Watch is a website that looks into retracted papers, and he's number 4 in their leaderboard. So that's, yeah. there's a couple who have even more retracted papers, but there's fabricators out there. So Diederik Stapel is definitely one of them. So now I'm going to switch gears, and I'm going to talk a little bit about my own research. So I'm specialized in looking at image duplications in science papers. So when I was doing my work on plagiarism, I was flipping through a thesis that had a lot of plagiarized text. But I was just curious, going through all these pages on the PDF, and suddenly I saw a blot, a little photo of a protein experiment that looked very similar to a photo I'd seen before on, in another chapter of that same thesis. But except it was flipped, like it was turned upside down. But it had a little smear, little smudge that I recognized. And so I'm like, wait, I've seen that blot before. Let's go back. And I found, I'm like, wait, it's upside down. That's weird. Maybe they made an error. But it was also representing a completely different experiment. And I was just, what's going on? And it turned out that this was also published as a paper. And so I delved a little bit deeper into the work of this graduate student and found more of these cases in which she had flipped the same blots. Like, there were 3 different blots that she kept on reusing in, all orientations you can think of. And it was supposed to show a different experiment. And so I thought, okay, that's weird. Maybe there's more of these cases. Let's just make this my new strange hobby. Let's just go to PLOS ONE, which is a scientific journal. Just go online. It's very easy to flip through all the PDFs that are published there. Let's just look, the first 100 papers and see if I can find examples of these types of image duplications. And sure enough, I think within the first 10, I already found 1 or 2. And so I was very motivated. Turns out that in the end, I did 20,000 papers. So this was like 4 years of my life. But I found about 4% of these, about 800 papers, that had duplicated images. And we call them, in our paper, we call them inappropriately duplicated. And by that I mean, like, you could, of course, reuse a figure to represent the same experiment. That's probably okay. But if you reuse it, if you flip it, or you say it's a completely different experiment, and you still show the same results, that is not okay. So that's inappropriate. So we found about 4% of the papers And I have to say also, we limited our search to papers that had photographic images. Things like line graphs or tables, I'm not trained or I'm not good in recognizing falsification. You almost have to sit next to the researcher to see that they falsify a table, right? I mean, there's statistics you can use to look for falsification in numbers. But for me, looking at a photo, it's fairly easy to see if there's duplications. Like I'm looking here at this room, like, oh, those 4 chandeliers look pretty similar to each other. So it's sort of a duplication of something that I see in my eye. So I did this for 20,000 papers, and we encountered 3 types of problems. So I'll show you some of the examples. But first, I'll show you an example of what is not image duplication. So these are true, honest, good quality, well, let's assume they're good quality, biomedical photographs of experiments. You see some macroscopic figures. You see some on the top right, those little black stripes are Western blots. So those are proteins that are run in a gel and then incubated with a monoclonal to see if a specific protein is present. And if you look at these images, you see that all these bands or all these cells or all these photos all are unique. Every band has a shape, like it's maybe like that, or like it has a little smear or intensity. Every band is unique, and that's how it should be. But if you look at these images and you see duplicates, while they're presented as different incubations or different cell types or different monoclonals, that they cannot be looking exactly the same. And yet I found those examples. So here are a couple of examples of what we call simple duplications. So it's the exact same photo used again to represent a different experiment. On the top left, I'm highlighting the ones that look identical in green. You see 2 blots that represent 2 different messenger RNA experiments, and they're looking exactly the same. On the right, you see a couple of, I think it's like bacteria that are streaked on a Petri dish or so. You see highlighted with the red boxes and with the blue boxes a couple of photos that look exactly the same, and yet they're representing different experiments. And on the bottom left, you see marked in red and blue a couple of microscopy images that look very, very similar to each other. I try always to say they look very similar to me because that keeps me out of trouble. If I say these look identical, I'm already making an accusation. But on the other hand, these simple image duplications could be honest errors. Like imagine you make a photo book of your vacation and you have 4,000 pictures of Hawaii, and you make your little photo book as a memory, and by accident you put in twice the same picture. One time on page 2 and one time on page 36. That can happen. It can be an honest error. You didn't do it by the time you get the book, you're like, oh shoot, this photo has been included twice. So a simple duplication like these could be an honest error. But then the second category is a little less likely to be honest errors. So these are photographs in which I see an overlap between 2 images. On the top left, you see 4 images of 4 different treatments with 4 different levels of radioactivity cells were treated. But you see that the top 2 panels have an overlapping area that are marked in green, while the 2 right panels have an overlapping area that are marked in blue, suggesting that at least 3 of these 4 images are actually taken from the same specimen. They're just, shifted a little bit, but there's an overlap. But it's not the same, it's not a different set of cells that were treated with a different set of radiations. It's the same cells. They just chose a different field that maybe showed fewer cells in one panel, in one area of that microscopic slide, and a lot more cells on the other end. But because they were a little bit stupid and they left an area of overlap, I could pick up on this. But I would not be surprised if the bottom left, the 4th one in which I cannot find an overlap, if that was also the same photo, because who knows. On the right, you see an example in which a blot was flipped, was mirrored. And on the bottom,
left, you see an example in which a gel was shifted and representing a different experiment, but it's the same bands that you see. Type 3 is even worse. This is where an image is photoshopped or altered in some way. The top graphs are fax plots, and they're, I don't even know how to explain it very well, but I recognize the plots because I've looked at 20,000 papers. Cells are sucked into a machine and tested for the presence or absence of two different markers. So on the x-axis is one marker, on the y-axis is another marker. A cell can be double negative, then they end up in the bottom left corner, or double positive, they end up in the top right corner, or they can be in between in one of the other quadrants. So where a cell ends in one of these four quadrants of the plot will tell you if it's double negative or positive for one of the two markers. But since every cell is sort of behaving like sand. If I had a handful of sand and I tossed it in a certain direction, and then I do the same experiment and I toss another hand in exactly the same direction, where the sand will end up in this room is going to be very similar, but the individual grains of sand are going to be at different positions. So these plots are similar. If you redo the same experiment, they cannot look exactly the same. They cannot have the dots in exactly the same positions. And yet these two graphs look super similar to me, except for the region in between the pink and the orange. There's a couple of regions that are different, suggesting that this plot was manipulated, like some dots were added or some dots were removed. In the bottom panel, you see that lanes 9 and 10 look very similar. All these examples are from papers that I found and that have been retracted. So it's not just my judgment, it's like all the editors and authors agreed that these papers should be retracted. So I've let you stare at some of these blots. So now it's time for some audience participation. So here is a panel of, 6 by 5 plots. They should all look different. They're all different treatments or different cell lines or different proteins. That doesn't really matter. They should all look different. And I want to see a raise of hands. Just stare at this. If you see at least one overlap, just raise your hand, and we'll let this incubate for a while. There's more than one overlap, I can tell you that. See some hands already? So you guys are hired. You can come. You can join my team. So I'll reveal what I see. So several of you saw some overlaps. So if I stare at these images for a while, I can mark them like this. So there's lots of overlapping areas. So do you believe that this is an honest error? No, I don't believe either, but you don't really know what's going on. Maybe it's a super sloppy lab and they didn't have much time to look at their photos and write a paper. But yeah, this suggests that it's more than an honest error. So like I said, I've done this for about 20,000 papers when we published our paper. Since then, I think I'll probably be at around 30,000 now, because I keep on doing this. So we found that 4% of these papers have inappropriately duplicated images. We also did some follow-up research. About 10% of these are probably going to be retracted. A lot of these are honest errors, and a lot of the times the author will acknowledge that there has been some mess up, but they'll have very weird excuses. And I'm trying to write a book or a list of weird excuses, which can vary, like, we made a mock-up photo and we forgot that we duplicated these images, or the student did it, and that's the most common one. It's always an intern or a student who gets thrown under the bus. But as an author on a scientific paper, it's not just nice to be an author on a scientific paper, you are also responsible for all content of that paper. So even if you are the professor in a lab and you claim you didn't know what your graduate student was doing, you are, as the professor and the senior author on that paper, you are responsible for what happens, and you approved this paper, and you did not see that the graduate student maybe messed up, but you're equally responsible for being on that paper. And if the paper gets retracted, then yeah, that's still a little bit your fault.
even though you might not have done it. And as a PI, as a professor in a lab, you actually have that responsibility because there's a lot of pressure on graduate students by their professors. And if you are a professor who says, I don't care, how you get this result, I just want your blots to be positive by Friday, what is a graduate student who is maybe on a visa and afraid to lose their job, what are they going to do? And so the pressure as put by a professor onto all their staff in the lab is also very important and can create an atmosphere in these where these things might happen. So about 10% of those, so that's 0.4% of papers will probably be retracted as the result of this. So far, if you just look at all biomedical papers, about 4 in 10,000 have been retracted as per numbers by the Retraction Watch, which I think means that about 90% has still been not discovered yet. But so I have my work cut out. So I actually I quit my job and I am doing this as a volunteer because somebody has to do it, and I hope I hope I can get by, but I saved some money so I can at least take a year off and keep on doing this. So we've covered science misconduct, but as I was in the past couple of months since I quit my job, I was just randomly going over papers and scientific literature, and I uncovered, or not really uncovered because many other people have already talked about this in scientific papers and on blogs. I've ran into a lot of examples of other bad science. And one of the examples that I'm most also obsessed about, I guess I get obsessed pretty quickly about particular things, but these are predatory publishers. So a predatory publisher is a publisher who claims they run scientific journals, who claim to do peer review, and who run under the open access model. So the open access model, for those of you who are not familiar, is sort of a fairly new phenomenon in science publishing, in which you, as the author, pay the publisher to get your paper published, and then the paper becomes free for others to read. Like PLOS ONE, for example, is an open access publisher. You can just go online. You don't need to work at an academic institute or have a very expensive subscription for that journal. You can just go online and read all the papers because the authors have paid the publisher to have these papers published. So it's about $1,600 or so, I think, to publish in PLOS ONE. But these predatory publishers make use of the fact of this open access model because they just care about an author paying them, and they don't care about the quality of the paper. They just also feed upon the pressure that a lot of people have to publish. And so a lot of inexperienced researchers might actually be falling for one of their spam messages that they send massively to all students and graduate students and postdocs and other scientists. These predatory publishers send emails like, please publish in our journal. We offer your peer-reviewed paper, and we're going to have a very fast peer review, and we're going to publish your paper. Just come to us and give us your paper. And so people might fall for these scams because it looks like it's a scientific paper. It's maybe called the International Journal of Microbiology, and you're like, oh, that sounds really good. And then you look at the publisher, but then you might actually find that these publishers have maybe hundreds of different papers on any topic you can think of: economics, agriculture, biology, medicine, psychology. They're huge publishers on paper, but then if you look at these individual journals, their contents are mainly empty, or they're filled with very, very poor quality papers. And they claim, even though they claim that they do science peer review, their papers don't look like any reviewer has ever looked at it. They're full of plagiarism often, language errors, weird and completely weird and crazy ideas. And in between, there's some science papers real science papers with real content, and they're probably written by inexperienced researchers. But once you publish your paper in one of these predatory publishers, you cannot republish it again, because that's the rule of science, like you can only publish things once. Unfortunately, these predatory publishers are not indexed in any of the search lists that most reputable
science get published in. So nobody will find your paper unless maybe you do a Google search or so, but you cannot find them by the regular way that scientists scan the literature with keywords. And so they're fake. They often have fake impact factors or even fake editors. My name might be somewhere on I might be a editor and I might not know it because they use names of scientists to make it look like those scientists are on their editorial boards, while in reality, they have not given permission to do so. And so a lot of inexperienced researchers might fall for these scams, but unfortunately, lone authors with wild ideas, as we all know, exist, because now it looks like they have published in a peer-reviewed paper. And also some shady companies, some industry scientists might actually misuse this as a platform to publish their not very well executed research that would have never gone through peer review in a real scientific paper. They might publish it there because now they can say to their general audience, oh, look, we have peer-reviewed papers, because the general audience does not know the difference between the International Journal for Microbiology. I'm just making this name up. I hope I don't insult if there's a real International Journal of Microbiology. But it's really hard for a layperson to know which papers are real and not real. One thing you can tell is, it's not necessarily a red flag, but very often it is the time between submission and publishing. So regular peer review, and I've peer reviewed hundreds of papers myself, that takes months. The whole process of peer reviewing and sending it back and then resubmitting it, that process cannot happen in 0 hours. That's like this, you can see here, this paper was submitted on February 23rd and magically accepted on the same day. So you can imagine that there's not a single reviewer has looked at this paper. And even more funny is that these predatory publishers also organize predatory conferences. So they are very happy to accept a couple of $1,000, and they'll organize a conference in a hotel room, maybe at the Hyatt Regency, but instead of organizing one conference, and as any conference organizer will appreciate, it takes months and many persons to organize one good conference. There are some, WASET conferences, organizes, if you look at their website, 53,000 conferences each year. But what they do, they rent one room in one hotel, and 200 conferences happen in that same room, because every conference maybe has 1 or 2 participants or 0. So in the end, only 20 people show up or so. And people actually submit fake papers to these conferences and go there and report undercover and then report back. And it's very funny because nobody understands the paper on a topic they are not interested in. And everybody in the end also gets an award, so everybody wins. And they're completely fake, but they're being misused because people like to travel to nice places. And if they can tell their boss, oh, I'm going to the International Conference on Microbiology, organized by WASET, can I get some money to go to Sydney or some other nice place that I want to go to? Of course, you can go to that conference and present your paper, and nobody will ever know you're actually sitting in a room with 20 people and listening to topics that you weren't interested in. So it's very easy to submit a paper to these conferences. There's a nice generator of scientific papers called SciGen, in which you can generate your own paper. All you have to do is give it your name of the authors, and it will generate a paper for you. So I wrote this paper with 2 of my cats, Patch Permeister and Oifa McFloof, which are my 2 cats. Well, one cat is a lone cat, but okay. And it's decoupling IPv4 from 802.11b in reinforcement learning. Now, and especially with the layout, you can create this in 30 seconds if you go to this website. You can create a very impressive paper to impress, I don't know, your parents or your neighbors. But it's if you read carefully the text, it's nonsense. The grammar is there, sort of, and there's a nice figure or so, but it's just all nonsense text, and you can just write it. If you submit it to these fake conferences, it will be accepted. And because so it really pays. I mean, of course, we're all skeptical here, but if you think it's fake, it probably is fake. So never stop asking questions if you read stuff like that. And finally, I want to touch upon some weird things I came across in the past couple of months. So people sometimes submit to real scientific papers, but they suggest a couple of peer reviewers who would be very nicely reviewing their work, and they give a list of names, but they all give email addresses that are actually made by the author, and so the author can review their own paper now, and of course you can imagine the peer review is very nice. And so these are other scams in which there's fake peer review rings. And I came across an interesting phenomenon that I didn't hear much about, which are peer-reviewed scientific papers, so published in real journals, not predatory journals, by people who seem to have very impressive affiliations, but they're fake, like they don't work there, or they have never worked there, or it's an institution they made up themselves. So on the top right, you see a guy, there was a recent Gizmodo article. This guy claimed to be a Harvard MD or PhD, depending on which article you read. He was featured a lot on many popular science websites. He was a psychologist who did research in things like suicide or like weird sexual things. So he was very popular and interviewed a lot. But it turned out he only did a summer internship at Harvard and never did a PhD there. He wasn't hired. And he wrote these weird papers, and he claims to work at some forensic, I cannot read my own slide, but like some institute he made up himself, and he said that he had ethics approval by that institute. But yeah, so he basically approved his own research on very weird topics. The guy on the bottom right, I call him the space dentist. He claims to do all kinds of experiments, and he has like 16 reviewed papers that I've now all reported to the journal, because I personally think they're all fake, because the results don't make sense. He claims to have worked at the Kepler Space Institute. I called them up, that's a real institute. And they said, oh yes, we know this guy, but he has never worked here. He does not work here currently, and based on your information, he will never work here. And so, and then the guy on the bottom left claims that he has a mathematical model for DNA, that females and males have differently polarized DNA, and that he can determine the gender of an unborn baby by infecting a pregnant woman with influenza virus. You either have to pick the male influenza virus or the female influenza virus, and you can then determine the gender of the baby. He claims to work at an Iranian institute. He has worked there, but he does not currently work there. They're very concerned, because I tweeted about this guy. They're very concerned about this. And he's been, like, while we're here at the conference, he's been spamming me, but he's very angry that I talked about this research, because he thinks it's real, and I think it's not. So with that, I'm going to leave you with a couple of, feel free to take photos, lots of websites that I love to visit and look at these types of research. But I want to stress again, most science is good. These are just some bad apples. But it's always good to be skeptical, and even at papers that are published in peer-reviewed journals. Thank you.
I think while we're maybe getting set up for Skeptics, we can take at least a couple of questions.
Great talk. I just wanted to make a quick point, though, that there are a lot of very reputable journals, including Nature now, that are expecting money to publish open access. So it's not just predatory ones. It's getting to be very common.
No, it's open access is being used, like I said, +1. It's just being misused by some of these journals. Open access papers by itself, but it's an important point to stress. An open access paper by itself is a good thing, because like for me, I'm not associated, so I actually have access to a lot of science now. It's just the model is being misused by particular shady publishers.
We'll take the next one at the microphone.
So I wanted to add, you had mentioned that there's a lot of predatory journals. I'm sure a lot of people in here are familiar with the Master Journals list on Clarivate Analytics that gives a list of scientific papers that are subscription-based, that are reputable, that universities subscribe to. Mm-hmm. And you can also use the impact factor to see like how big of an impact the papers have had. Is there something comparable for spotting predatory conferences?
Yes, and that's actually on this list. So that's Beale's list. It's now on bealeslistweebly.com. So you can go there and check if a journal is on that list. It's declared to be predatory. Obviously, some journals are in the gray zone, and they might not agree with being included in this list. But if they're on that list, they're probably predatory.
What about for conferences?
There might be a list, but I would not know. I mean, I usually look up, I don't know, WASET conferences, and you'll find tweets about it, like this is spammy, or this is... Just going online and seeing that they organize thousands of conferences that are all happening in the same week in the same hotel, that's a huge red flag.
Thank you.
It's as a scientist who gets those emails, it's relatively obvious. If you're looking for it, if you're aware that predatory conferences are a thing, not always obvious, but you can kind of spot it.
Yeah.
This might be out of your purview, but I was just wondering, what do you think of the things like the so-called hoax, or the guys that submitted that paper called The Conceptual Penis or something like that, where they're trying to basically scam a journal? Do you think that's in your bailiwick, and do you have any thoughts about that?
Yeah, that falls under the category of false papers. I've come across a couple of those. I mean, I think it's funny, and it just exposes the journal maybe as not being very diligent in really looking at their papers. And so if it's obviously funny, like some of those examples, I don't really care about it. I'm like, okay, well, that's fairly obvious. But yeah, some of them fall in between where you're not quite sure if it's funny. The mathematical DNA, I actually thought it was a joke. And then I'm like, okay, this guy is really serious. He was decapitating birds as well. And like, okay, that's beyond where it's still funny. And but it is, yeah, there's gray zones where people make up things, and it's just not clear if they're just trolling or really are convinced that that's true.
We're going to take the last question at the microphone here, and that will be it for this speaker.
Well, I just wanted to say that I'm very surprised that there aren't more free automated solutions for checking on whether both the images, because that's not a hard thing to do these days, and the text aren't plagiarized. I wonder if you could comment on why that hasn't happened.
So plagiarism in text is actually, that check is being done by most reputable journals. They have Authenticate running, they probably pay a lot of money for it, but most journals check at some point either before sending it out to the peer reviewer or after acceptance. They check their papers. So it's really hard to do plagiarism nowadays. The predatory journals obviously don't do that, and they don't care. Images is much, much harder than most people think. I've had tons of people coming up like, oh, this should be very easy to solve. I participated with my data set in a DARPA challenge with artificial intelligence and whatever, and all kinds of words I didn't understand. But I had the data set, and that's been 2 years. Nobody has been able to crack the code yet. But there's some promising leads. It's very, very hard to find these things because they're not exact matches. It's just a pattern I recognize. But if you look at the pixels, they're not always completely identical, and it's really hard. Having said that, this will be, in a couple of years, we'll have solved this problem, and I don't have to do this anymore, but we can rely on those things. I'm sure the crooks will come up with other solutions to fly under the radar, but we have to deal with this now.
Thanks.
Sure.
Please give Elisabeth another round of applause.